Okay, right. In this video, we are going to look at the second application of genetic engineering, and that is the production of insulin. Okay, let's do some revision. Do you remember insulin? It is an important hormone which controls the blood sugar level in our body. Okay, back in Form 4, we have learned about the concept of homeostasis in regulating blood sugar level. In the older days, insulin was extracted from the pancreases of cattle or pigs to treat diabetes mellitus patients. But nowadays, insulin can be commercially produced by genetic engineering for patients that suffer diabetes mellitus. Besides insulin, other successes of genetic engineering includes hepatitis B vaccine, blood clotting factor, and growth hormone. Okay, this photograph here shows the photo of E. coli, Escherichia coli, which serves as the cloning vector in the process of producing insulin. Now, okay, let's look at this figure. Shows how human insulin gene is transferred to a bacterium by genetic engineering to produce insulin. Okay, so number one, a spliced human DNA with insulin gene. Okay, and here we are a plasmid, which is a circular DNA found in bacteria, which is used as cloning vector. So this insulin gene is cut with restrictions enzymes. Okay, for plasmid, the plasmid is cut with restriction enzymes as well. And here we are, this is the insulin gene and also a cut plasmid. So these DNA fragment and also the plasmid, they are cut with the same restriction enzymes that works like a pair of scissors. And the enzyme is the DNA ligase enzyme. Okay, next is that this is now a recombinant plasmid, which is successfully inserted with the human insulin gene. Okay, so we call this thing as a recombinant plasmid. And this is a bacterium. Usually, this cloning vector is E. coli in this case. And the recombinant plasmid just now is introduced into a bacterium. And after that, this bacterium is called a transgenic bacterium, or in other words, this bacterium is genetically modified bacterium. Okay, so bacterial clone multiplies. Okay, so the E. coli multiplies and copies of insulin's genes are also produced. Okay, so large amounts of insulin are made by protein synthesis. Okay, so the last step is that insulin is then extracted from the transgenic bacteria and purified for use.